I know that because I number my lectures. And this will be my last lecture for a few weeks because then we're going to start with our guest speakers. And they're going to go into sort of reaching out into related areas for biological rhythms in health and disease. But today we're going to talk about sleep. I have worked on the edge of the sleep field for a number of years. We're not really doing that so much right now, but I have participated in sleep meetings. I've been in this part of the sleep advisory board to the government and for the Sleep Research Society, and I've spoken to clinicians on the biological basis of rhythms in sleep. So this is sort of, the talk I'm going to give today is something that's kind of come out of those experiences. And I hope it sort of gives you some insights on how you spend about half of your life. That's right, I have a video on that today. I suspect I'm not going to get through everything I have, but I'll get through enough to get you the foundations, basically, of what you need to know to hear about what some of the other speakers. And then, is it plugged in? And then what we'll do is at the very end, there's a video, a very short video that I want to make sure that we go through. So at some point, I'm going to make a decision about where we are and how we're going to proceed. Maybe the lights just burned out.
Can we function with the computer?
We can function, but Harry, would you just run next door to the micro office and tell them? And they probably have a spare.
Okay.
Just who's ever in there at the desk. That'd be great. So sleep. I bet you guys think you probably don't know very much about sleep, but I'm here to tell you you already know a lot about sleep. And let me give you an overview of what we're going to talk about first. How and why do we sleep? Uh-oh, what happened? Oh, tell Harry that we don't need any help.
We wanted it on. No, it's good. We wanted it on.
Yeah.
It's too cute.
Who knew there was a second switch? Anyway.
We got it, Harry.
Thank you, Diane. So we're going to talk first about what is sleep and what are the processes that it regulates, or that regulate it. How can alteration in these processes cause sleep problems? How sleep affects brain and body functions, and then the fact that you really do need sleep. And most of us live with some sort of sleep debt every day, and we catch up on the weekends. Wiley Post was the first aviator to try to fly around the world, and he did fly around the world. He thought he wouldn't have any trouble because he would stay with the sun, and being in the presence of the sun all the time would permit him to stay awake. But he hadn't been out very long until he'd been up about 20 hours and he realized he had this extreme urgency to sleep. And that's because sleep is an endogenous process. It isn't strictly driven by the environmental presence of light or darkness. And then what are the consequences of sleep need and sleep debt? So what is sleep and what are the processes that regulate it? And I have this movie, I don't know when it was made. Some of you, a couple of you have seen it before. But it's, what's kind of cool about it is its observations on sleeping people. And it gives you a sense about, it's not a new movie. I don't know exactly when it was put up on the internet. But it's made by well-known sleep researchers. But it's going to give you, wait a minute, I think it should work. It's going to give you a sense of what it looks like when you're sleeping. Let me see if I can get the little, here we go. It has this great music. Oh, and it was done in another country. Tell me if you can hear it well enough. Can you hear it?
In the course of an average life,
Should I make it louder?
we spend between 20 and 25 years asleep.
I have to make it louder by doing this. Luckily, it's about, I'll make it even louder, then we'll go back. It's louder this way.
life, we spend between 20 and 25 years asleep. But scientists don't know why.
This mystery has perplexed us since the beginning of time. Imagine this, spending 1/3 of your life and not knowing why you do it and what its purpose is. Sleep is one of the greatest mysteries that science has struggled with.
Now, as scientists develop the ability to look inside the sleeping brain, they may be on the brink of a remarkable discovery. It may be that the most important function of sleep is to allow us to dream. In the 1950s, researchers working in the newly emerging field of sleep science began to measure electrical activity generated by the sleeping brain. They soon discovered that sleep was not, as had been previously believed, a time when the mind lay dormant. It turned out that, in fact, the brain went through a series of transformations during sleep. As we fall asleep, our brainwave recordings change. As wakefulness begins to slip away, we become drowsy. This is stage 1 sleep. Gradually, we descend into deeper levels of unconsciousness, stages 2 and 3 sleep. Then, after around 20 minutes, we reach stage 4, the deepest form of sleep, in which the brain lies dormant. Scientists call it slow wave sleep because of the slow undulating brainwave recordings it produces. But then, after 90 minutes, the brainwaves suddenly change. Frenetic, jerky movements appear, indicating a burst of mental activity.
It is an awesome experience. I remember the first time I saw it, I just couldn't believe it. You see these slow, rhythmic patterns, just disappear. And it looks as though the person is awake. The person is sound asleep. And the only thing that you see is jerking movements of the eye muscles. And that's what was called rapid eye movement sleep.
It's hard sometimes if you don't see the eye movements themselves. If you just look at the EEG, the brain waves, you can't tell a REM state from a waking state. It looks just as active as if you were wide awake.
Periods of rapid eye movement sleep emerge with clockwork precision, at 90-minute intervals throughout the night. During these REM periods, the mind is more active than when we're awake. And when the early sleep researchers began to wake people up during the different stages of sleep, the reason for these sudden surges of brain activity became clear.
If you awaken somebody out of stages 3 or 4, if you ask them what they were thinking, they'll tell you that they were thinking nothing. They were, as far as they could tell, very deeply asleep. REM sleep is different because that's when we do our dreams.
Michael, wake up. Can you tell me what's been going through your mind now?
Yeah. I was at a circus and was laughing at a clown in a big red suit.
If you wake somebody out of this state of sleep that we call REM sleep, most of the time they'll report a dream. Awaken them out of the non-REM stages, and most of the time they will not report a dream. So there's all this complex thinking going on that the person is able to relate when you get them out of the dream.
That's all I think.
Okay, thank you. You can go back to sleep now.
Sleep
Real to real.
researchers rely heavily on our waking memories of dreams. Though it's possible to measure electrical activity in the sleeping brain, no one has yet invented a way to record the dream as it unfolds. But even if these dream reports are made as soon as we wake, they can only ever be a distant reflection of sleeping thought. As we emerge from sleep, the chemical composition of our brain changes rapidly. And for most of us, the dream slips quickly away.
One of the other fascinating aspects of REM sleep, which I think is clearly understudied or underappreciated, is that you have no memory of the event. You wake up from your REM sleep state and the memory of what was in your brain is vanishing, which is very unique. It's different from wakefulness. It's one of the most important differences from wakefulness. You are inventing things. It's totally new images and things that are not real, but these images disappear very quickly after you wake up.
Dream sleep has another characteristic which sets it apart from the other stages of sleep. In deep sleep, though our brains are inactive, it's nevertheless possible for us to move our bodies. Those who sleepwalk do so in deep sleep. But in REM, we're paralyzed. The only muscles we can move are the ones in our chest, which enable us to breathe, and the muscles of our eyes. It's called sleep paralysis, and we all experience it every time we enter REM. Sleep paralysis is a protective mechanism, nature's way of making sure we come to no harm when we dream. Its existence is evidence that evolution has gone to enormous lengths to preserve the dream state. But why should dreams be important to our evolution as a species? And when does dreaming begin? Babies spend about 9 hours a day in REM sleep. But no one knows when humans start to dream.
We have no idea when they begin to dream. We know they have very heavy REM time. We don't know when the neural networks are set up and connected well enough to sustain that kind of visual imagery with the storyline. But we know that some infants certainly scream bloody murder at night as if they were having a nightmare long before they could tell us about it.
In the womb, the fetus spends most of its time in REM. Though perhaps because it doesn't need protection, sleep paralysis hasn't yet developed.
The majority of the state of the human organism in utero prior to birth is dream sleep. All the kicking and twitching that a pregnant mother feels during the last third of the pregnancy is assumed by most people to be wakeful movements, but in fact, these movements are the twitches of REM sleep.
REM sleep is not confined to humans. It's been identified in all species of mammal, even the largest.
Do animals dream? I don't know, it's hard to question them, to ask them. But they do have the physiological capabilities as we understand it. You might surmise that something is going on, because physiologically it's identical to what we have. You see the same brainwave patterns, you see the same eye movement patterns, you see the little twitching occurring in your dog or your cat.
REM isn't even restricted to advanced animals. The duck-billed platypus has 6 times more REM sleep than humans. It's the most primitive of all egg-laying mammals. It coexisted with the late dinosaurs. This means it's possible that even the dinosaurs had REM sleep. But to know whether dinosaurs dreamt, we'd have to be able to ask.
Okay. There's some very, I like that video for a number of reasons. It gives you a sense of sleep as a behavior which I have put up there, that it's an active state, which people didn't used to think. And it introduces you to some of the concepts about how sleep has been analyzed. And when they had the chart with the many pens moving, that was measuring electrical activity at the surface of the brain, the so-called electroencephalogram, or EEG. People still use that. They use all digital measuring and recording devices now. But they still are very interested in what's going on with respect to this macroscopic measurement of sleep states. We still don't know a lot about REM, although I assigned you a paper which was from Giulio Tononi's lab, which was one of the real steps forward in trying to understand what's going on during REM sleep, and we'll talk about that a little bit later. But let's go ahead. Some of the issues are still with us. Many of the issues are still with us, and the sleep field, it's a very passionate field about trying to understand this very, all systems behavior, and then what goes wrong when you have sleep disorders. So, universal characteristics of sleep-like states is the absence of voluntary movements. In other words, you can't decide to reach your arm up because you're not really aware of what's going on with your extremities. It's spontaneous, and it occurs in a circadian rhythm, which simply means it's there with about a 24-hour period, and it's there in constant conditions. It's reversible with sensory stimulation, and that's as opposed to pathological states such as coma or exhaustion, where you might appear to be sleeping, but you're kind of just in a recovery state. So sleep is reversible. You can go in and wake somebody up, but your arousal threshold is increased, and so you have to go in and actively shake people, give them a very loud noise, a very kind of alarming noise, and then you can wake them up. The characteristic species-specific posture and resting place that minimizes sensory stimulation, so in other words, keeping away from stimuli, is characteristic of all species during sleep. So in other words, you tend to, most animals tend to curl up like the elephants were. Cats, dogs, they tend to minimize the exposure of the soft body parts to the outside world, of course, because their vigilance is down, but also it's conserving temperature. So one of the things a sleeping individual has to worry about out in nature is that they can't really control the temperature in their environment. So they try to get in a place, in a posture that their temperature is preserved. And it's a homeostatic mechanism, that is, it has a sort of steady state aspect to it. So you come back to a certain baseline as time goes by. And you see sleep-related changes in neural functions, and that includes this decreased sensory input. So in other words, you're gating out the sensory systems so that visual input, auditory input, tactile input have a higher threshold and are less likely to raise you out of sleep. And it's an identifiable species, stable species characteristic, and that is many, many, many species of animals, including, I believe I put it at the top, 19 species of fish have been studied, and sleep has been identified, reptiles, amphibians, and invertebrates such as the worm, the fly, the cockroach, and the octopus, which means it's pretty universal. And these characteristic features are preserved or seen in some form in all of those organisms. So, the two-process model was developed by a Swiss sleep scientist named Alex Borbet. He's originally Hungarian, and he determined that his model is based upon the idea that there are two regulators of sleep. One is time of day, and that's the circadian process, labeled as C here, and the other one is called the S process S, and that is the process where you use something, your energy, some sort of necessary force in your life up when you're awake, and you need to restore it when you're sleeping. So in other words, sleep is a balance then between an active organism and a period where you need to be inactive to recover what you need to be active again the next day. And that's the process S, or so-called process sleep. And the model going to talk a little bit more about the model as we go along, is during the waking period, the circadian process is oscillatory, and then it's low at night when you're sleeping. But the process increases over the course of the day, and then at the transition between wakefulness and sleep, it changes direction and goes down, and this would be the restorative phase, and then it starts to rise again with waking the next day. So this is based upon, he and Irena Tobler studied many species, but they mostly worked on humans, may I say. And then we see that the S phase of sleep is manifested in individuals that are sleep deprived. And so here you have slow wave sleep, which are those early phases of sleep that they saw in the electroencephalograms, where you have basically from the very active, chattery trace that you see during wakefulness, you start to see sort of more synchronization in what the neurons that are being measured at the surface of the brain are like. And so you see these rhythmic waves, and this is over the number of hours of sleep. And these dark bands are the periods of REM sleep, which start, as they said, about 90 minutes in, and then the duration of REM increases over the course of the night. So just before you wake up, you tend to have your most consolidated bouts of REM sleep, and that's when you also do most of your dreaming. So then what happens if an individual has been sleep deprived for a couple of days? There's a number of characteristic differences here between these two. This would be the normal situation. First of all, we see that the sleep duration is much longer. So this is someone who's really been, stayed up long periods of time. The first thing that happens is that you sleep longer, presumably recovering during that recovery period, recovering what it is that you missed because you weren't sleeping for some period of time. Oops, wrong. And then you notice that the number of REM bouts is increased, and they continue to increase again over, this person was up 13 and a half hours. Notice the amplitude of the slow wave is higher, and this continues till about several hours into the night, and then you get this sort of regular alternation, but many more cycles of it than you saw under normal conditions. So this is part of the evidence that there's a recovery or homeostatic aspect of sleep. Obadiah.
Is there any evidence that there's a difference in the quality of REM sleep for later stages than earlier bouts?
You mean in the deprived individual? Or just here?
Just in the normal one.
Yeah, in the normal one, you do get more REM toward the end of the night. But you mean more REM bouts?
No, better quality REM bouts.
Oh, better quality? I can't answer whether it's better quality. I don't know if people, what the assessment of quality is. Because they're usually just measuring REM by the electroencephalogram pattern being wakeful-like, and basically indistinguishable from daytime-like. And then with the rapid eye movement, and they can even measure those with a little, muscle sensor up on the eyes. But as far as the quality, probably the thing you'd have to do there is wake people up. And they do that, and ask them, what's going on. They can also do it after they wake up to see how they perform on learning tasks and various other kinds of cognitive tasks. So, the model has evolved a little bit, and these Durkheim Dyke and Stephen Lockley are some of the major people working in this field now. And basically, what I'm going to show you now is the primary regulators of sleep and how they interact, but then the sort of secondary regulators, of which there are many. So we have the clock, which is the time of day signal, telling the organism that it's nighttime in the outside world. The clock is in the night state. It's sending night state signals out to the rest of the body, and one of those signals is going to the brain, telling the brain it's time to sleep. And the other one is this homeostat that's basically restorative, and this is peripheral to the SCN. The homeostat is not in the SCN. And we know that sleep-wake cycles feed back on the clock as well as on the homeostat. So you have this sort of interacting oscillatory system that we've talked about in my earlier lectures. Now, what are some of the other, how do these interact? So the model then is that the clock provides an alerting signal during the daytime, and this rises during the night. Have any of you ever worked really late and you sort of start to nod off, and then you sort of make yourself become awake again? You can actually do this, and once you do that, if you miss a certain sort of magic period, you can stay up a long, long time and be much less tired than you were during the period right about here where the alerting signal peaks. And then notice there's a shift in this alerting signal. That would be something that's keeping you awake. And it opposes, the model is that it opposes this homeostatic drive for sleep load, which simply means this, I've inverted this pattern now, but that while you're awake, you accumulate something, that's your sleep debt, that you then reach a switching point where these two signals interact, so the forces that are driving you to go to sleep because you're tired, basically, overcome and counteract the alerting signal of the clock. And how do we know, or what is the basis of this model? Well, characteristically, we lesion the SCN. When you lesion the SCN, the question is, what happens to this sleep pattern? And what you can see is it's no longer consolidated, which we see in most things that are circadian in their patterning. But we see very short duration, low amplitude oscillations in sleep and wakefulness over, this isn't even a day here, that they've measured it. And so there's really a change in the patterning of sleep, and this is simply showing you a model based upon some experimental data. But what it doesn't tell you is also the quality of what kind of restoration this pattern does for you. Now, the regulators of sleep are more than just the sleep homeostat and circadian rhythms. But we have social time as a regulator of sleep. How many of you, I know it's not fair to ask students how late they normally stay up because you're kind of driven by need, whatever you have to get done. But do any of you stay up later on Saturday night than you do on Thursday night, for instance? That would be, especially if you're out with your friends, or you go to a late movie, something like that. And that's a form of arousal that can actually feed right into your sleep-wake cycle and allow you to stay awake late and also to go to bed, get up late in the morning. And that's an adjustment based upon these arousal signals that are coming in, usually at the end of the day for someone in their real world. This is often seen in a great study that was reported from Spain. It was a study on the sleep-wake patterns of feral Spanish students, which simply means they're students out there in the real world. And what they found is they had a fairly regular pattern of getting up, especially in the morning, getting up for those early classes, and then their going to sleep pattern got to be a little bit later on some nights. But as soon as Friday and Saturday night came, they stayed up late. I mean, we all do. Late on Saturday morning, got up later Saturday night. Late on Saturday night, get up late on Sunday morning, but then you have to go back to Monday, whether you're a student or you have a job, and you have to be able to shift back. So this is an interaction of social time with this system. And then we also have the light-dark cycle through the circadian photoreceptors, the melanopsin, retinal ganglion cells we talked about last week. And this is feeding into the clock and adjusting time of day. And interestingly, melatonin, that hormone of darkness, which is produced at night when it's dark, is regulated by the clock, because that tells the pineal gland that it's night. You can go ahead and make melatonin. And the light-dark cycle, because it can only be made when it's dark at night. And so melatonin is a regulator, is regulated by both of these, and it also regulates, melatonin can feed into the sleep-wake cycle. Now, we see that sleep patterns change over our lifetime. They told us in the video that prenatally, the infant spends most of its time in REM sleep, so not only is it sleeping, but it's mostly in REM sleep. It's interesting to think about why that might be. But then in a young adult, this is the way that sleep staging is normally presented based upon those waves from the electroencephalogram. So this is stage 1, 2, 3, and this is very deep slow wave sleep. And then we come up a couple stages and jump up to REM. They're putting REM at the top in this way of presenting it. And then notice after about 2 cycles, about 3 hours into sleep, there's an arousal. That's the magenta star is an arousal period where the person wakes up briefly, and then it goes back into various stages of sleep, and here's another complete awakening. Here's the increase in REM sleep. Now compare this pattern to that of an elderly adult. Same number of hours asleep, basically, but look at the fragmentation you see here. Very little, very deep sleep, very little even stage 3, but much more oscillation and alternation between the lower sleep states, and then lots of arousals. These are all arousal points. There is REM sleep, but it isn't showing the same pattern it showed up here, where it tended to get longer as the night went by, but these are all fairly short. Here's the longest bout of this whole period. So very fragmented sleep, and this is typical of the elderly, and in fact more of their sleep intrudes into the daytime period. We see more napping and so forth. Now we can then add age into another regulator of sleep, so that the very young and the very old have input from their life state. It can be hormonal, it can be deterioration or maturation, but these age then can also feed into the sleep-wake cycle. Oh yes, and then we have my favorite one, volition, and that simply means choice. You may choose to stay up late, and we all do that. And so if you're motivated enough, you can stay up really late and study. If you're motivated enough, you can watch an all-night movie. I've done that. Or work on finishing a grant or meeting a deadline. But you can actually choose to overcome these forces that are normally keeping you on your rest activity cycle. And of course, I should add, volition can also be your job. It may also be that you have to work at night. And so regulation of sleep is complex, and interfering with the normal process by some of these mini inputs can also have health-related consequences. And that's, I'm going to say a little bit about those, but we also had, I selected a second paper which I thought was a great sort of state of the field of sleep with respect to sleep health and sleep wakefulness. Oh yeah, I should get big. So what are the processes that regulate it? They're circadian and homeostatic. There are intrinsic processes, light and dark and social experience. They're the so-called ontogenetic, that is over the course, on the genes, over the course of lifespan, age, and then voluntary sleep curtailment. And voluntary sleep curtailment is really the basis of many of the most common forms of sleep disorders. So, why did I put this all back up? We don't need that now. So, how are the processes that regulate sleep related to sleep dysfunctions? Now, I have the slides here that I had from my previous talk, where we talked about sleep disorders arising within the circadian system. You guys all know that it can. I told you the story about Louis Patachek's family that went to bed very early, the advanced sleep phase syndrome. And I was telling people, it was the prospective job candidate, about this at dinner the other night, because she's very interested in genomics, and people didn't realize that there were actually human mutations in transcription factors, which is what the clock genes are, because there's a whole debate over whether transcription factors aren't so important that you can never change them. But I said, oh no, in the circadian clock protein transcription factors have many different forms in humans, and they are the basis of diseases. So we're just going to go right past this now. And I talked about the differences in the casein kinase 1 epsilon. But now we're going to move and talk about sleep disorders arising within the homeostatic system. And this is Emmanuel Mignot's dog story. Now, Emmanuel Mignot was the Frenchman in the story. I almost stopped to tell you. He's just a delightful, very gentlemanly Frenchman. He's been at Stanford for most of his career, but he's originated in France. And he sees patients, he's a neurologist, patients with sleep disorders, but he's a big dog lover also. And I've had dinner with him several times. His pet dogs are all dachshunds. But he has a line of Doberman pinschers that he's maintained in his lab, because these Doberman pinschers have a very specific defect. And that is, when they are aroused, either they see you coming down a hall and they get all excited, or if there's a loud noise, they immediately collapse. They immediately fall on the ground and sort of like they're passing out, and then they'll get up again. Do any of you know what human disorder that dog was developed to reproduce? Where arousal during the daytime, so it's not when you would normally be sleeping. It's called narcolepsy. Narcolepsy. So it's not uncommon in the human population. Now, there was a grad student when I was a grad student who had narcolepsy. I've known people whose children grew up to have narcolepsy. Tends to be more Southern European. At least that's one group. There probably are others now that they've studied it more generally. But Emmanuel Mignot's guiding light in his scientific research part of his career was to discover the gene for narcolepsy. And I remember this so clearly. I was at a marine biological station one summer where my husband was working, and it came over public radio. The gene for narcolepsy had been found. And they started to tell the story about Masumi Yasunaga, who's at UT Southwestern Medical School, and that he'd found this peptide that when you knock the gene out, these animals had this behavior where, if these were mice, of course, if they were in the cage and they decided they had to go over and have a drink of the water bottle, and someone went up to sniff them, that animal would fall over. And they were looking, the gene that they knocked out, which they did on purpose, was a gene they thought had something to do with eating. It's a gene that they predicted would affect their eating behavior. But it didn't really have much of an eating phenotype. That is, it didn't have any manifestations of sleep disorders. However, what they did was something very smart, which I'm telling you because you might be able to use it someday, and that is they just decided to put a video camera on the cage and monitor these cages. These were group-housed animals. They probably wouldn't have seen it so clearly if they'd been singly housed. These mice, when they got aroused, knocked over. Well, bumped over. Meanwhile, Emmanuel Mignot had been doing this the hard way, where he'd been doing gene mapping on the chromosome. And that used to be the way people figured out the genetic basis of disorders. It's very laborious because you basically study the chromosome physically, you look at what genes map close to it, and then you select breeding to kind of narrow down the spot on the gene. So within a month, Emmanuel published his paper, and he had a different gene that was causing this narcoleptic behavior in the Doberman pinschers. His was for the receptor for the peptide that Yanagisawa had found. Yanagisawa had found the orexin peptide, and it's named for like anorexia, where you don't eat. Orexin was thought to have something to do with eating, and he named that peptide. Meanwhile, the group at Stanford had found this peptide very, very abundantly in the hypothalamus, and they named it hypocretin because it was only in the hypothalamus. And these turned out to be one of the same peptides, and now basically orexin is the term that everybody uses. The receptor that the dogs have been bred to lose was the hypocretin orexin receptor 2. So it was one of the peptide receptors for this peptide. And that actually is unbelievable evidence that this gene is critical in mice and rats for narcolepsy. You always want, especially if it's a signaling molecule, to be able to regulate the signaling molecule and look at the consequence, and then to look at the very specific key or receptor that molecule interacts with to activate the signal transmission. And that is really an incontrovertible proof that you're dealing with that particular signaling pathway. People have looked at this gene in humans. It is not as simple in humans. This gene, having a defect in either of these genes alone, is not enough to make you narcoleptic. I have not heard recently what progress they've made in figuring this out. You can be treated for narcolepsy. Usually this has got like a late teenage onset, but you know, you got it the rest of your life because it's basically in your genes. So there is a great interest in finding a way to treat narcolepsy in ways that doesn't interfere with other parts of the affected individual's lives. So this then developed, especially things like narcolepsy where you can have these animals awake or asleep, it seeded the idea with Cliff Saper, who is an alum of Illinois and received a Distinguished Alumni Award, and I might have mentioned this earlier in my lectures, I can't remember. He's at Harvard Medical School, and he developed the notion of the flip-flop switch relationship between sleeping and waking states. And this is the basis of, it's sort of the neural basis of that model that I showed you where we had the circadian arousal signal and the sleep homeostatic signal coming together. And so his notion is that then in the base of the brain where the SCN resides and in nearby nuclei, we have interactions between two other nuclei that determine whether you're awake or you're asleep. And this includes the orexin neurons, which are in the hypothalamus, and the ventrolateral preoptic neurons, which are also in the hypothalamus. And that these feed in, the orexin would have an arousal signal, right? Because if you don't make the peptide or you don't have the receptor, you tend to fall asleep. And so these would feed into parts of the brain that are involved in sleep regulation, the locus ceruleus, the tuberomammillary nuclei, and the raphe. Raph's got the serotonin, that's an arousal signal. And you are then, when this signal's feeding into these regulatory nuclei, you're awake, and you're inhibiting the ventrolateral preoptic nuclei. So that would be the wake state. And you can see if you have a key regulatory region and it's fed into by two different brain regions that have opposing effects, that you could then establish a switch relationship between them. And then in the sleep state, you would turn off the activation step, and then the VLPO, or ventrolateral preoptic nuclei, would inhibit. Not only do you stop producing the activating signal, but you actively inhibit this region. You actively inhibit the orexin neurons, and then this is permissive for sleep, which involves many other brain regions. It's not just that your hypothalamus is the only part of your brain that changes during sleep. So this is the model, and this model has been very well received, and I believe is currently the state of the thinking of the field here. Now, I'm going to talk a little bit about genetic variations in processes that regulate sleep that can lead to different kinds of sleep dysfunctions. Oh yes, the question becomes, how much sleep do you really need? And that's a question that you should all ask yourselves after you hear the rest of what I'm going to talk about. Because, you know, we tend to compress our sleep into the least sleep that we can get by with sometimes. And that's sort of the way of the modern world. The question is there a consequence of that? You know, how much sleep do you need to get by and do a really good job? How much sleep do you need to get by and do an adequate job? How much sleep can you not operate without? And there are people who really seem to need a lot of sleep just to be able to, you know, operate in the daytime, and others are pretty good at beating it back and keeping it low, but they may not be operating at their best. So sleep affecting brain and body functions. So here's our interaction between the two again. And the sleep-wake transition then marks the point at which that model switches from wake active to sleep active. Sleep is for the brain and for the body. So now I'm going to say a little bit about what goes on in the rest of the brain. during the time you're sleeping. So during awaking, your neocortex, basically, the more surface parts of your brain, show this desynchronized activity, which is why you see these very irregular patterns on the electroencephalograms. And there's very high glucose utilization. That's because active neurons need a lot of energy to support the pumps that reverse the ion fluxes that take place when neurons are active. So neurons are active because ions flow through channels on their membrane. And there are other processes that need energy, but those need the most. And so it's a very highly energy demanding stage of your 24-hour behavior. And then during non-REM sleep, the neocortex shows the synchronized activity and glucose utilization goes down. Glucose utilization goes down. Now it becomes a very interesting question is when you start to see synchronization and you get the waves emerging that are characteristic of especially the deeper sleep states, what's mediating that? What's permitting those neurons' activity to become synchronized? And then what's rapidly reversing it up here? So whatever's happening, it's very rapidly reversible. So then, oh, we saw that. So then, okay, this is what I want. So then, basically, if we go into this is a preserved human brain, and this was a slide produced by Bob Moore, which I've modified. This is where the parts of the brain we're talking about lie. So this is these are the wake nuclei in the neocortex, and the thinking is that the homeostatic sleep drive, that process S that you're using up when you're awake, arises here as well as in the VLPO, which is that part of the brain that was involved in the switch to sleep. And the major neurotransmitter the major neurotransmitter in the VLPO is an inhibitory neurotransmitter called gamma, or gamma aminobutyrate. And so it is the universal inhibitory signal in the brain. Then it innervates these regions that are in the hypothalamus. This is the mammillary nuclei and preoptic area and so on, down into the brain stem. And these areas are also involved in sleep. I talked a little bit about the cholinergic parts of the brain as well. And so this inhibitory area has to feed all the way down and regulate the rest of these nuclei by turning them off. And this process, this inhibition by the VLPO's projections, is critical both to the onset and to the maintenance of sleep. And so if you have damage to this very specific area in your hypothalamus, you will definitely have persistent dysregulation of sleep and lack of coherence of sleep. Now, they mentioned in the little video that sleep is universal, and it's seen in very, very ancient animals like the platypus. But the question is, with respect to REM sleep, the part of sleep that's involved in dreaming, and the thinking is consolidation of experiences to form long-term memories based on your previous day's experience. It's very different in different organisms, and no one knows why. And so, here you have some very primitive organisms. I believe the possum is considered pretty primitive. The bat isn't so primitive, but the hedgehog, you can just tell by the way they look that these are primitive organisms. And they don't have, there's a whole range from 8 hours to 3 hours of REM sleep. Human, yeah, 2 hours of REM out of 8 hours of total sleep. And then over here you have low REM animals, the guinea pig, the baboon, and actually larger animals, including the cetaceans, the sea mammals. And so these are very generally short REM animals. They're living lives in a complex environment. Why is REM shorter for them? We don't know the answer to that question. And this is what the sleep scientists love to think about. What's different about the habits of these animals, the environments that they experience, the lessons they have to learn that makes REM so different. And that is still, to the best of my knowledge, not known. And then this is related to the paper that we're going to be talking about. This was one of the first pieces of evidence that sleep could facilitate memory consolidation, and that it had something to do with memory. And that is, they took our little friend, the hooded rat, and they trained it on a specific maze. They trained it on the same maze several days in a row. And then they looked at activity in its hippocampus, the part of the brain in which memories certainly begin to be stored. That's sort of the input site for memories that have to do with learning about space, which is the space around you, which is what the hippocampus is known for. And what they saw in this animal is a sequence of activations that looked very much like what they saw when it was taking the learning the maze. So that gave them the idea that during the sleep phase, it was retracing the paths that it had taken while it was learning the maze. Now, what's great is to get some people working on learning and sleep together in a room, because there are the people who say this only takes place during REM, and REM is the only important aspect of sleep for learning. But there are others who attribute learning to the non-REM phase of sleep. That probably means, the best explanation for that is that we know there are different forms of learning, that there are contributions to learning by different phases of sleep. But the current most interesting work is coming out of people like Tononi's lab, where they are showing that, in fact, that it's REM that's necessary. So, oh right, what processes peripheral to the brain are regulated by sleep? So I talked about sleep involving changes in the brain, but it also involves changes in the body. And these have to do with hormone profiles and endocrine function, metabolic function, and we know that our metabolism changes between day and night. Our liver enzymes change. The later you eat, the more likely you are to have lots of lipids in your blood during sleep, because you're not metabolizing the same day you would have at noon. You see changes in the immune function related to sleep and sleep deprivation. And then there are cardiovascular variables. And this is putting everything together then with respect to hormone secretion. And so we have the sleep-wake homeostat and we have the clock. Both of these relate to and regulate the autonomic nervous system. Now, how many of you guys know what the autonomic nervous system is? Auto that tells it all. It's things you don't have to think about. Heart rate, blood pressure, body temperature, basically the organ functions that are controlled basically by the state of things that's integrated in the brain. Both sleep feed into that and the SCN, but also the hypothalamic endocrine system. And when we say pulse generators, it simply means that the projections from the hypothalamic brain nuclei down into the pituitary gland, which is the major neuroendocrine factory in the body, that then can release hormones directly into the bloodstream as the blood's going through the pituitary. Those hormones tend to be released in pulses, so you have very large amounts released, and then it's released in waves. And so the pulse generators go down to the pituitary, and they cause many different hormones to be released. Some have to do with sexual behavior or state. Some have to do with stress, like cortisol. Some have to do with growth hormone, thyroid hormone, prolactin. So in other words, many of the sort of state-changing hormones that go into the periphery. Now, it's interesting to me that the whole body system is organized so that you have time of day regulation as well as sleep state regulation. For instance, growth hormone is only made at night. It's only made at night, and it's made in very high levels soon after you go to bed. It's why you want to make sure younger children, where they're really using that growth hormone to get those bones long, have long, healthy sleep bouts. Stress hormones change during the night, and we know that from one of my first lectures where I showed you there's a time of day at which heart attacks were most common, and that's right before dawn when your cortisol levels rise. So they're low at night when you're sleeping, and then they rise the next morning. So another really very interesting area, and this kind of brings us toward the metabolic aspect of the rhythms in health and disease, and that has to do with signals that regulate food intake and energy balance. And these involve two peptide systems. And we know a lot about these two peptide systems now. So peptides, of course, are small amino acid sequences. Many of them can diffuse quite a long way from the site at which they're released. Many of them can get into the blood and, like vasopressin, and be distributed throughout the body. One of them is called leptin. Leptin can act at hypothalamic sites, which simply means there are leptin receptors there. There are receptors for leptin on the muscle cells and on the fat cells. And with insulin, refeeding means eating, exercise, and hyperglycemia, that means when you have lots of energy, you inhibit food intake. So basically you can see how leptin is acting. It's a signal that you should stop eating. What do you think leptin levels are going to be like at night, since we're talking about leptin in the context of sleep? Should be high. That's a sort of a control that you're not going to be inappropriately taking in food.
They should be dropping right before we wake up, though, right?
Right.
Kind of opposite of cortisol.
Right. Because what do you do when most people get up?
I'm hungry.
You're really hungry. And then the other peptide that really works in antiphase to leptin is a peptide called ghrelin or ghrelin. I usually pronounce it ghrelin. It counterbalances leptin in the hypothalamus, so it can go into those cells or regions that leptin can act on. It also has peripheral effects that depend upon the tissue, so it can have variable effects in the periphery. With cannabinoids, fasting and hypoglycemia, that's low blood sugar, it basically is motivating you to eat, and it rises before meals, specifically to stimulate food intake. So here you have two peptide systems that are perfectly poised in the ways in which they work, and then the modifiers, like in this case with insulin and exercise and so on, to make sure they work at the right time, in the right context. So this then is basically a map of, this is from Eve van Cauter, so this is in humans. It's the feeding hormones, leptin, which you could also call the satiety hormone because it's inhibiting food intake. It's like after you've eaten Thanksgiving dinner, no matter how much you like pumpkin pie, you can't eat anymore because you're sated, your leptin levels are high, and then you have a ghrelin, which is the hunger, the drive for hunger peptide. You now, if you look at them, they undergo diurnal variations, and here's a pattern for an individual human where the leptin levels are high at night, and then, just as Sam predicted, they start to go down before morning, and you're not in a room where you're smelling food. During this period, you're still sleeping. But past the middle of the night, the ghrelin levels start to fall off, and then notice here, where are they rising? Right after, in this case, right after you ate. This one's rising a little earlier. And then toward the end of the day, they start to go up. It might even make you think that the later you eat, the less you'll eat because of your leptin levels are starting this circadian rise. And here we have the ghrelin, and this would be positive for appetite then. And notice that it's starting to rise during the night. It's fairly high in the middle of the night, and it starts to go down. But then right here, it starts to rise again just before you're eating breakfast, rises well before you eat lunch, and well before you eat dinner, and then it's very, very low at the time the leptin levels are rising up. In fact, they're almost in antiphase in this early part of the night. So, you know, you don't get hungry before a meal only because your stomach is empty or your blood glucose level is low, but also because your ghrelin levels are starting to rise, which is cueing everything else that it's time to think about eating. So, this is a transition now. Let's see here. Does sleep loss impact the processes that sleep regulates? Well, of course it does. Otherwise, I wouldn't be putting that up there. We looked at sleep deprivation. In this case, we're going to look at individuals from a study by Beth Clearman and Dirk-Jan Dijk. These people were at Harvard Medical School when they did this, which has a great big human sleep group. And basically, what we're looking at, the rate at which people fall asleep depending upon their habitual bed rest duration. So basically, they're taking people in, and they're basically saying, you know, how long do you usually sleep? And the people, and I believe this was self-scored with short duration, slept less than 7 hours, relatively short duration sleep. They brought them in to the lab. They put them in a very comfortable, easy chair, very low lights. They let them just sit there, and they asked, how fast do they fall asleep? And what you see here is their data, the mean time of sleep onsets relative, yeah, this is sleep onset, relative to the duration. And notice how fast the people with short habitual bed durations fall asleep. It takes for them to fall asleep relative to those with sort of medium, which is with about 8 hours. And then those with 9 hours, you know, they can basically sit there for hours in the dim light in the daytime and not fall asleep. And then this is the mean for everybody. But this is why some people fall asleep in seminars in the afternoon. You know, same kind of environment, especially if the seminar isn't extremely interesting. So this is an indication that people may choose to sleep short periods. They may even tell you that they only need a little bit of sleep. But if you put them in this environment to test their duration to falling asleep, they fall asleep rapidly, and this is an indication that they're sleep deprived, especially when you look down here at these people who sleep for a longer period of time. And these are simply some cognitive tests that the group gave them, and I'm not going to go through them in great detail. All I want you to know is that the amount of time you sleep affects, this is your first nocturnal sleep opportunity. This is again telling you fall asleep really fast. That's sort of a summary of what I just showed you, and showing that the different stages of sleep show the same basic curve. So the people with really short durations all tend to go up and show more time sleeping if they've had a longer period to rest, longer time to falling asleep. This is the one where they're looking at their alertness and performance. And in this case, they gave them several kinds of tests. One was simply a visual test, that's this one. One of them, they had to calculate, you know, simple math problems and see how fast and accurately they could calculate them. And this is tracking lapses, and this is reaction time. And what you can see, if you just look at the red, in each case, if people came in after their short usual sleep duration, and then they gave them an extended period of 12 hours of sleep, all of them got better or faster. This is good when you're going down because your reaction time is better. And that's another indication that these people were somewhat sleep deprived and they weren't able to perform at their optimum when they were under their short sleep period. So this gave rise to quite a large group of studies looking at the effect of the relationship between sleep, short sleep, and sleep disorders. And this is simply showing you, summarizing what I did, that sleep duration appears to matter, especially with respect to cognitive deficits. But the interesting thing is these people are self-imposing sleep restriction or wake extension, you can look at it either way, in their choice of lifestyle. All of us do this. This is what all of us do. I do it all the time. And the question is, to what extent is it affecting our performance? It may be that you can perform completely adequately, but perhaps not in all cases. I guess that's a safe thing to say. And I'm not going to go into all of these right now because, oh yeah, that's a good. That's just lots of things change with your ghrelin states. And this I showed you. This one I have to show you. This is a video showing you the 24-hour transglobal travel. So in other words, each one of these yellow spots is an airplane. And that we're going to see, this is the shadow of night coming across the Earth. This has been sort of unfolded, and this is daytime. And what I'm now going to show you is the pattern of basically transglobal flight, and think about what it has to do for sleep for the people who are basically working in that environment. Now I have to find my, yes, there we go. My little arrow. Oh no, oh no. It's going to be so sad if I can't get this to work. Why wouldn't it work? Well, I'll tell you what. After the break, I will show you this video. But what is so impressive is you can see how the patterns of flights are organized to basically for the Western world, first of all, and to make life easy for the traveler to the extent possible, and put sleep pressures on the pilots. And I have that somewhere else on my computer, so I will find it and get it up. So this is simply showing you that there's lots of sleep curtailment, and I like to end with asking how much sleep you really need. And I will get the video working while we break, but great. I covered most of what I wanted to cover today, so we will now come back and talk about the papers. Thank you.